Bismillahirrahmanirrahim and Assalamu alaikum. Welcome to Biosciences with Shatin Swati. And Shatin Swati is here to deliver the next lecture from the bioenergetics. And this lecture is about light independent reaction, which is also known as dark reaction. And this is the second step of mechanism of photosynthesis. I have discussed first part of the mechanism of photosynthesis, which is light reaction, in detail in my previous lecture. So must go to my previous lecture, which is about the light dependent reaction or photophosphorylation. So before starting my lecture, I have a very humble request to all of you. Kindly subscribe to my channel, promote my channel, like my videos, share my videos. If you are sharing my videos, my lectures, you are sharing the knowledge. So keep watching, keep sharing knowledge with other. Okay, now I am going to start my lecture, and in this lecture I will discuss about the discovery, introduction, enzymes involved in the dark reaction, and at last I will discuss in detail about the steps of Calvin cycle. Okay, most of the students confused whenever I ask a question that what is the dark reaction. Mostly students replied that ma'am, the dark reaction is that reaction which occur at the night time. No, I will tell you, dark reaction is not that type of reaction which occur at the night. Why it is called the dark reaction? The reason is that this reaction do not depend directly on the sunlight. It take the energy from the light reaction in the form of the ATP and NADPH. So that's why it is called the dark reaction, because it occur in the stroma and do not take the sunlight directly and take the energy which is synthesized during the light reaction in the form of ATP and NADPH. So must watch my first lecture about the light reaction, in which these products synthesize, and then these products which are synthesized in the form of ATP and NADPH in the granum of the chloroplast, then move towards the stroma for the continuation of the other process, which is the Calvin cycle, which I am going to discuss in detail here. So first of all, I will discuss about the discovery. That is very important to know who discovered, because I'm mostly believing to to what to tribute all those scientists who worked and spent their lives in their discoveries and making the life and all the process easily understandable for all of us. Okay, the cycle was discovered in 1950 by Melvin Calvin and the James Basham and Andrew Benson at the University of California, Berkeley. By using the radioactive isotope carbon 14, as we know that carbon dioxide involved in photosynthesis, carbon dioxide and water, so that's why the carbon 14 was used by the Calvin, Andrew Basham, and Andrew Benson's and the James Basham, and for which he was awarded the Nobel Prize in 1961. And you can see the photo of Melvin Calvin on the screen. Okay, now as we are familiar with the equation of photosynthesis from our early childhood, we learned this uh, equation from, I think so, first second class, and this is the chemical reaction in which 6 carbon dioxide molecule and 6 water molecule react with each other and to form the glucose. C6H12O6. Glucose is a sugar carbohydrate in the presence of sunlight and chlorophyll, and producing the oxygen. So the step related for the production of the oxygen I have discussed in the light reaction. Water molecule during a light reaction undergo the mechanism of photolysis, and during photolysis 2 molecule of water produces 1 molecule of the oxygen. So in this reaction you can see here the 6 carbon dioxide when react with the 6 water molecule and produces the 1 carbohydrate and 6 carbon oxygen molecule, which liberated in the atmosphere, and sugar carbohydrate used by the other living organism as a food. So this is the overall mechanism of photosynthesis. As you can see on my right side, the granum in which chlorophyll is present and the light reaction occur in the thylakoid membrane and synthesize the ATP and NADPH. Then this ATP and NADPH move towards the stroma. This is the granum and this is the stroma. I discussed in detail in my previous lecture, so must watch that lecture, and then this will be easily understandable for you. Okay, so ATP and ADPH, which is synthesized during the light reaction, move towards the stroma, and this ATP and ADPH then utilized during the Calvin cycle for the production of the glucose, sugar. Okay. Now, what is light independent reaction and how will you define? So note down my lecture. The light independent reaction or dark reaction of photosynthesis are chemical reactions that convert carbon dioxide and other compounds into glucose, carbon dioxide and water into glucose. These reactions occur in the stroma. So here just I'm showing you the light independent reaction, so carbon dioxide and the water reaction occur during the water conversion occur during the light reaction. So carbon dioxide and other compounds include the ribulose biphosphate, which I will discuss later on, into glucose. These reactions occur in the stroma, the fluid filled area of a chloroplast outside the thylakoid membrane. These reaction takes the products ATP and NADPH of the light dependent reactions and perform further chemical process on them. The sum of the chemical reactions in the Calvin cycle is as following. Like 3 carbon dioxide, you can see here, requires 6 NADPH, 9 ATP molecule, 6 NADPH molecule required for converting 3 molecule of carbon dioxide into glyceraldehyde 3-phosphate. Must keep it in your mind. And as a result, NADP, ADP, water produce and inorganic phosphates. So the reactants, or you can say the substrate of the light independent reaction is 3 carbon dioxide. One cycle requires 3 carbon dioxide, 6 NADPH. Must keep it in your mind. If you have to solve the questions related to that or any MCQs, you can write. One cycle, 3 carbon dioxide, 6 NADPH required, and 9 ATP molecule. And these 9 ATP and 6 NADPH synthesized during what? Light reaction in the thylakoid membrane. Is it clear to you now? Okay. So this is the overall reaction. 3 carbon dioxide and 9 ATP and 6 NADPH required to produce that. Okay. Now enzymes involved in the dark reaction or light independent reaction, which is also known as the Calvin cycle, is operated by 11 different enzymes that catalyze 13 reactions. But the key regulatory enzymes are: number one, RUBP carboxylase. RUBP means ribulose biphosphate carboxylase. Fructose 1,6 biphosphatase, FBPase. Pseudoheptolase, pseudoheptolase 1,7 by bisphosphatase, which is known as SBP. Pseudoheptolase 1,7 bisphosphatase, SBBPase. RU5P kinase and NADP glyceraldehyde 3 phosphate dehydrogenase. These are the most important enzyme, 5 key important enzyme. 11 out of these 11, these 5 are very important key enzymes of the Calvin cycle are light independent reaction or dark reaction. Is it clear? Okay. Now, what is Rubisco? That is the very important, you can say, the enzyme. And what is Rubisco? Rubisco is the ribulose. Ribulose, the word ribulose means ribose sugar. And ribose sugar, you know that monosaccharide which consists of 5 carbon. Clear? Ribose sugar. And what? Ribose consists of how many? Carbon atom? 5 carbon sugar. And ribulose means attached of alcoholic molecule. 1,5 biphosphate. At one end, this is carbon number 1, this is the carbon number 2, carbon number 3, carbon number 4, carbon number 5. At carbon number 1, you can see here 1 phosphate is attached. And at the carbon number 5, you can see here the 1 phosphate is attached. So, this is 1,5 biphosphate. Is it clear? Why it is called ribulose? Because it is synthesized from the 5 carbon sugar. So 1,5, 1 and 5 biphosphate carboxylase oxygenase. Now, what is carboxylase and why it is oxygenase? This enzyme acts both as a carboxylase as well as oxygenase. Means oxygenase, it become oxygenase during photorespiration, which I will discuss in my next lecture. As an oxygenase, I am showing you here. This is the photorespiration. When the amount of carbon dioxide decreases, it become oxygenase, and when the amount of the carbon dioxide is normal, then it acts as a carboxylase during photosynthesis. Is it clear? Okay, so that's why this is the carboxylase oxygenase enzyme, commonly known by the abbreviations Rubisco, RUBPcase, RUBPco. You can pronounce it. Is an enzyme involved in the first major step of carbon fixation. Carbon fixation is the first step of Calvin cycle, and this is very important enzyme which help in the carbon fixation to carbon dioxide fixation to ribulose biphosphate compound, which occur in the stroma. A process by which the atmospheric carbon dioxide is converted by the plants and other photosynthetic organisms to energy-rich molecules such as glucose. Is it clear? Okay. So this is here. Rubisco act as a oxygenase because the concentration of oxygen is high, concentration of carbon dioxide low. Here, concentration of carbon dioxide high, so it become the carboxylase. The concept clear now? Okay. Now the Calvin cycle. Calvin cycle consists of 3 steps. You can see here on the screen. The three important steps of the carbon cycle. First one is the carbon fixation, in which the carbon dioxide fixed to the RUBP, ribulose biphosphate, with the help of enzyme Rubisco, carboxylase. The second step is reduction. Here the reduction will occur, reduction of the glycerate into glyceraldehyde. And the third step is the regeneration, means the regeneration of the RUBP, ribulose biphosphate. Because this RUBP occur in the stroma, synthesize in the stroma, and then regenerate. Regenerate, synthesize in the stroma, and then use during the Calvin cycle for the production of the food molecule, or you can say sugar, or you can say the glucose, carbohydrate. Is it clear? Okay. Now, this is the simplest Calvin cycle. As you can see, this is the first step in which 3 RUBP, ribulose phosphate. How many ribulose phosphate? 3 RUBP, ribulose biphosphate. This is the compound. That compound react with 3 carbon dioxide molecule. Must keep it in your mind. How many ribulose biphosphate? 3 RUBP react with 3 carbon dioxide molecule. In the presence of the most important enzyme Rubisco, carboxylase, RUBP carboxylase, because here the substrate is RUBP, ribulose biphosphate, so the enzyme name is related to this RUBP carboxylase, because it act or you can say convert the carbon dioxide, so that's why this is known as carboxylase. So 3 carbon dioxide react with 3 molecules of RUBP, and you can see here RUBP is the 5 carbon compound. How many carbon are present in the RUBP? 5. So 3 molecules mean 15 carbon here and 3 carbon here. Understand? 3 molecules and 3 molecules are 5 carbon. So it's mean 1 molecule contain 5. So 3 molecules contain 15 carbon here. 15 carbon here and 3 carbon here. So when 3 carbon and 15 carbon means the 3 molecules of RUBP and 3 molecules of carbon dioxide, when react with each other, it produces first the unstable compound, which is not mentioned here, which is 6 carbon. 6 carbon and 3 molecules of the unstable compound formed here. Then this unstable compound, which is 6 carbon compound, which is unstable, which is not still named by the scientist, maybe in the coming time it will be named. So the 6 carbon compound, 3 molecules of 6 carbon compound which are unstable produce here. Then this 6 carbon compound converted into what? 6 molecules of 3 phosphoglycerate or phosphoglyceric acid. Phosphoglycerate is also known as phosphoglyceric acid. The concept is clear now. Now you can see here the 6 molecules of the 3 phosphoglycerate. Now phosphoglycerate or phosphoglyceric acid is 3 carbon compound. Now here 18 carbon. So how many molecules of the phosphoglycerate will produce here? 6 molecule. If you multiply 6 with 3, you will get the 18 carbon. 6 3s are 18. So how many molecules of the phosphoglycerate produce here? 6 molecules. Because 3 molecule carbon dioxide, 3 molecule RUBP, when react, then they produce the unstable compound, then this which is 6 carbon, then this unstable compound converted into phosphoglycerate. Phosphoglycerate or phosphoglyceric acid is 3 carbon, and how many molecule will produce? 6 molecules. Is it clear? Now this is the carbon fixation. Now the next step is reduction. Now what will happen? Reduction of the phosphoglycerate into phosphoglyceraldehyde. What will happen here? glyceric acid is changed into aldehyde, phosphogylyceraldehyde. Now what will happen? Here the 6 ATP molecule converted into 6 ADP molecule, and 6 phosphate releases. Is it clear? And 6 NADPH converted into 6 NADP, and 6 protons or hydrogen ion releases here. And these 6 hydrogen ion or proton then react with this phosphoglycerate and convert this phosphoglyceric acid into phosphoglyceraldehyde or phosphoglyceraldehyde. Is it clear? That is known as GP. Now here also the 6, because these are the 6, so the 6 phosphoglyceraldehyde produce here, and these phosphoglyceraldehyde is 3, also the 3 carbon. Now what will happen? This is the reduction, because hydrogen ion or protons react with this phosphoglycerate and convert it into aldehyde or phosphoglyceraldehyde. Now this phosphoglyceraldehyde, which is also 3 carbon compound, and 6 molecule are produced. From these 6 molecule, 1 molecule move away from this Calvin cycle and converted into glucose, carbohydrate. The remaining 5 molecule, now 6, from this 6, 1 phosphoglyceraldehyde move away from that cycle or quit from that cycle and converted into glucose. Now how many remaining? 5 molecules remaining here. Concept is clear? 6 out of 6, 1 move from this Calvin cycle out, and remaining 5 molecules then react with 3 ATP molecule and produces again the RUBP, 3 RUBP. Now here 6 remaining, from 6 this phosphoglyceraldehyde produces glucose. Remaining 5, which are 3 carbon molecules, phosphoglyceraldehyde, this glyceraldehyde, which is 3 carbon and 5, so 5 3s are 15, and these 15 carbon containing 5 molecules of phosphoglyceraldehyde converted into 5 carbon ribulose biphosphate. Is it clear? RUBP. Now the 5 molecules of 3 carbon phosphoglyceraldehyde when converted into RUBP, which is 5 carbon, and produces 3 molecule. 5 3s are 15, 5 3s are 15. Is it clear? So RUBP is here again regenerated. The concept is clear now. Then this again undergo the Calvin cycle and react with other 3 carbon dioxide, and cycle is repeated again. Is it clear to you? Must keep it in your mind and count the carbon dioxide carbon, RUBP carbon, and then you can clearly understand the concept or the chemical reaction, balancing of all the chemical equations. Like this is the phosphoglycerate, which is 3 carbon. This is the phosphoglyceraldehyde, which is 3 carbon. So 6, 6, and out of 6 phosphoglyceraldehyde, 1 move out of that cycle and produces the glucose, and remaining 5 molecules, which are 3 carbon, produces the 3 molecules of ribulose biphosphate, and this ribulose biphosphate, which is 5 carbon, again react with 3 carbon dioxide molecule and produces this. So I hope the concept will be clear upon you people. If you have any question, write in the comment box. I will try my best to solve your problems, your questions. I will be back. Thank you very much, and I will deliver the next lecture about the chlorophyll So keep watching my lecture, like my videos, share my videos. Till then, Allah Hafiz.